Let us now understand how the gut bacterial metabolism affects the digestion of resistant starch. The bacteria in the lower intestine of humans may be wide open to as much as 20 grams of resistant starch per day. The fermentation products of resistant starch by gut bacteria include gases like methane, hydrogen, carbon dioxide, and short-chain fatty acids, which are mostly acetate, propionate, and butyrate. Much lesser amount of organic acid, branch, short-chain fatty acids, and alcohols are also produced. Starch degradation is a supportive process in lower gut, degradation of starch polymers in glycolysis with short-chain fatty acids or organic acid as an end product. Methane production by methanogenic from formate, hydrogen gas and carbon dioxide products of bacterial metabolism of resistant starch. Short-chain fatty acids. Short-chain fatty acids are the metabolic products of anaerobic bacterial fermentation of polysaccharides, oligosaccharides, protein peptide, glycoprotein precursor in the large intestine, including those derived from dietary fiber and resistant starch. The principal short-chain fatty acids are butyrate, propionate, and acetate, although other short-chain fatty acids are also produced in lesser amount. Short-chain fatty acids are preferred respiratory fuel of the cell lining of the colon, that is called colonocytes. They increase colonic blood flow, lower luminal pH, and help to prevent the development of abnormal colonic cell population. In addition, it can affect gene expression and may induce cell cycle arrest or even apoptosis of naturally programmed cell death, which removes DNA damage, unwanted or old cells of colonocytes. Therefore, dietary intervention that increases the amount of short-chain fatty acids in the colon are thought to be beneficial to the gut health, and short-chain fatty acids are commonly used as a marker of fermentation and colonic health. The length of transit time and diet are the two variables known to influence the concentration and type of short-chain fatty acids found in the colon.
A long transit time result in protein breakdown and an increased contribution by amino acid fermentation to short-chain fatty acid pools, while dietary fiber, including resistant starch, can modify large bowel and fecal short-chain fatty acid levels.
Let us now understand the physiological effect of resistant starch. A number of physiological effects have been attributed to resistant starch and are presented in table. Resistant starch, by evading digestion in small intestine, has few interactions with other constituents of upper gastrointestinal tract. It is fermented in large intestine, resulting in marking of such fermentation products such as carbon dioxide, methane, hydrogen, organic acid, and short-chain fatty acids. Let us now understand, as shown in the table, the physiological effect. The first effect, improved glycemic and insulinemic response. The condition where it may be productive effect like diabetes, impaired glucose, insulin response, and metabolic syndrome. Second is improved bowel health, which can be found in colorectal cancer, ulcerative colitis, inflammatory bowel disorders, diverticulitis, and constipation. Improved blood lipid profile, that is usually helpful in cardiovascular disease, lipid metabolism, and metabolic syndrome. Prebiotic and cultural protagonist which is helpful for colonic health. Increased satiety and reduced energy intake is useful for obesity. Increased micronutrient absorption, which is good for enhanced mineral absorption and helps in preventing osteoporosis. Along with that,
adjunct to oral rehydration therapy.
It is useful for the treatment of cholera and chronic diarrhea. Synergistic interaction with other dietary components like dietary fiber, protein, lipids and thermogenesis, which is found to be useful for improved metabolic control, enhanced bowel health and of course obesity and diabetes. Let's conclude. Resistant starch, the share of starch and starch product that resist digestion, appears to deliberate several health benefits. It may discuss substantial benefits to human colonic health but appears to have less an impact on markers of lipid and glucose metabolism. Comparison between the studies are vulnerable by poor experimental design and differences in type and doses of resistant starch used. It is likely that resistant starch intermediates some or all interest in increasing its regarding its prebiotic effect. At present, resistant starch is included under the umbrella of Institute of Medicine of National Academies and AACC definitions of dietary fibers, and it is measured under the remit of AOAC method for analysis of dietary fiber and labeled accordingly. Further research imports to include more in-depth examination of effect of resistant starch on colonic function and inflammatory bowel disease. There is also necessity for properly designed control studies to determine the exact effect of resistant starch on human lipid and glucose metabolism, particularly over the longer period of time and in individuals with impaired glucose response. The possibility of resistant starch as an agent in weight loss and maintenance regimes will certainly be explored as a part of tries to curb global increasing incidence of obesity. As yet, the exact mechanism action of resistant starch remains unknown and more studies are needed to confirm whether these effects are mediated via short chain fatty acids or prebiotic effect.